Scientists from the University of Cambridge have created model embryos from mouse stem cells that form a brain, a beating heart, and the foundations of all the other organs of the body. It represents a new avenue for recreating the first stages of life. The team of researchers, led by Professor Magdalena Zornica Goetz, developed the embryo model without eggs or sperm. Instead, they used stem cells, the body's master cells, which can develop into almost any cell type in the body.
This study shows that you can use a special device that we have made and special media that allows to grow natural mouse embryos.
By guiding the 3 types of stem cells found in early mammalian development to the point where they start interacting, the researchers mimicked natural processes in the lab.
To use the same system, and we can put stem cells that are grown in the dish for a long time. We can put them in these conditions and they self-organize alone over 8 days into a whole embryo.
The scientists were able to get the stem cells to talk to each other by inducing the expression of a particular set of genes and establishing a unique environment for their interactions. The stem cells self-organized into structures that progressed through the successive developmental stages until they had beating hearts and the foundations of the brain.
We'll be focusing on taking stem cells, skin cells from a patient. Let's say I'm a patient, I need liver cells, and take a biopsy, make stem cells in the dish, grow them in special condition, and put them in this device for about 20 days, and they will make organized structure, which we call synthetic embryos.
They also had the yolk sac, where the embryo develops and gets nutrients from its first weeks, unlike other synthetic embryos. The Cambridge developed models reached the point where the entire brain, including the interior portion, began to develop. This is a further point in development than has been achieved in any other cell-derived model. According to the team, the results could help researchers understand why some embryos fail while others go on to develop into healthy pregnancy.
And they have cell progenitors, including liver progenitors or blood progenitors, and we can take these cells maybe and transplant them back to the patient.
In addition, the results could be used to guide the repair and development of synthetic human organs for transplantation. The study, which is the result of more than a decade of research, is progressively led to more and more complex embryo-like structures. Was reported on August 25th, 2022 in the journal Nature.